The overall goal of this procedure is to measure contractility of tracheal smooth muscle from mice. This is accomplished by first preparing the tissue baths and calibrating the isometric force transducer. Next, the mouse trachea is isolated and mounted on the force transducer rods. Then, the trachea is stimulated and the resulting tension is measured and analyzed. Ultimately, results can show contractility of tracheal muscle through isometric force measurements.
The main advantage of this technique over existing methods like in vivo measurements of airway resistance is that in vitro tension measurements provide a more direct assay of smooth muscle contractility. The disadvantage of this technique is that it is an organ bath preparation, and therefore circulating factors that may affect smooth muscle contractility in vivo may be absent in the in vitro preparation. Though this method can provide insight into mouse tracheal smooth muscle, it can also be applied to other smooth muscle tissues, such as large vessels, various gut, and bladder smooth muscle where ring preparations of muscle are sufficiently large that they can be mounted on force transducer wires.
Two components of the contraction measurement device require some description. They are the tissue bath and the force transducer. The tissue bath maintains an oxygenated physiological solution at a warm temperature. For mice trachea rings, a 10 milliliter tissue bath is used, which is equipped with a water jacket for warmth, glass inlet to bubble oxygen, and inlet and outlet ports to change solutions. Stored away, there is a reservoir of PSS solution, bubbled with oxygen and maintained at 37 degrees Celsius by a water bath.